All right, guys, what's up? We're going to be talking about an exciting topic called protein synthesis. Okay, that's going to be good stuff. Now, what we're basically talking about is how does DNA actually create something? Okay, we know the structure of DNA, we know it's the code of life, but how does it actually create something in the body? And what DNA creates is protein. So, open up to a new page in your notebook, title it Protein Synthesis, and these are extremely important notes. Okay, prior knowledge. So don't write anything. We're just going to take a look at what you should already know, and if you don't, then we'll talk about it here. Okay, so what is protein synthesis? It's a fancy, pretentious way of saying we're building proteins in the body. Proteins are made up of smaller molecules called amino acids. So if you have some chicken for lunch, we say, oh, we get a lot of protein from chicken. But realistically, your body is going to break all that protein down into amino acids, and then your body will build it back up depending on how you need that built back up. DNA contains the code, therefore controls the cell's production of proteins. All your body's processes are controlled by proteins. Okay, so go ahead and write those two. Last things down. So write this down. DNA contains the code, therefore controls the cell's production of proteins. So that's how DNA actually is the brains of the cell. And all your body's processes are controlled by the proteins. So your DNA doesn't control the body directly. It simply tells you what proteins it can make. And then write this down. Ribosome, this is the place where the proteins are assembled. So what are ribosomes? Ribosomes were those little tiny organelles. They were just like a little dot. When we made our poster of the cell, they were just a little dot. Sometimes they were on the rough ER. Sometimes they were just floating around in the cytoplasm. So they're really, really tiny organelles. So pause the video, write those last three things down, and I'm going to move on. All right, RNA. Remember, don't write anything unless I tell you to. Want to make sure your notes are concise. Okay, RNA. So kind of like DNA, stands for ribonucleic acid instead of deoxyribonucleic acid. It is the workers, quote unquote. Takes instructions from the DNA, uses it to build proteins. It's a single strand, so DNA is that double helix shape. RNA is not. It's a single strand. It's got a different sugar than DNA. Its sugar is called ribose, and it has different bases. So this is kind of interesting. Instead of having A, T, C's, and G's, it has, instead of thymine, it has this uracil thing right here. So keep that in mind. U will pair with A with RNA, but not DNA. There are three types: mRNA, tRNA, rRNA. Now you don't need to take any notes on this page, but the next page you do, because we're going to go through and discuss what the three types of RNA are. All right, types of RNA. First, you have messenger RNA. The messenger RNA is extremely important because it is what's going to actually bring the DNA's instructions from the nucleus to the ribosome. Okay, so that is important. That message has to get out of the nucleus somehow. Transfer RNA. This brings amino acids to the ribosomes to be assembled. Okay, so these tRNA molecules with amino acids on them are just floating around the cytoplasm, around the ribosomes, and those are going to be extremely important for actually banging the substrates, which are the amino acids, to the ribosomes to build a protein. Last, you have ribosomal RNA. And the best way to describe ribosomal RNA is it's just your ribosomes. Ribosomes, we call them organelles, but theoretically, they're just a massive enzyme that allow the messenger RNA and the tRNA to meet up and create proteins. So write everything you see on the screen down. Go ahead and pause the video if you need to, and I'm going to move on. Okay, so there are two steps to protein synthesis, and you need to know each step, where they happen, and what happens. So let's simplify the first step: transcription. So transcription is, here's how I simplified its definition. Write this down. mRNA strand is created from a gene found on the DNA strand. So we're taking the DNA strand and we're going to a specific part of that strand and creating a messenger RNA strand or an mRNA strand. So that's the simplest way you could define the process of transcription. So write that down. Then we're going to go on to a detailed description of it. So when you think of transcription, your mind needs to go, okay, we're making mRNA from DNA. But let's go ahead and get you some better notes down. So how does this happen? Well, it happens in the nucleus because that's where the DNA is. And you are going to write everything you see on the screen down, but let's talk through it first. So DNA helicase, an enzyme, unzips the DNA. We know the DNA is 2 strands, so to be able to get our enzymes in there, we have to unzip it. And we'll do some practice with this, so don't freak out about any of this kind of confusing stuff. So free RNA nucleotides pair with complementary base pairs. So basically, A goes to U, C goes to G. Just like with DNA, if you did any of that work, then we know that A's and T's line up and C's and G's line up. One side of the DNA is copied. RNA nucleotides bond together to form one mRNA strand. Just like amino acids make up proteins, RNA and DNA are made up by nucleotides. So these are just, the nucleotides are the A's, U's, C's, and G's, and T's. Okay, those are when you see nucleotides, it's just talking about the letters that make up RNA and DNA. All right, the mRNA strand leaves the nucleus and heads to the ribosomes, and the DNA strand reconnects. So we just described the process of transcription in detail. So go ahead and pause the video and write those down. All right, so let's take a look at what that looks like. You obviously don't need to draw this or anything. Just pay attention. Go ahead and pause your TV that's playing in the background. This is important. So what goes on here? Well, like we said, we have to unzip the DNA strand. We see here's the DNA strand out of a chromosome here. DNA is usually not in chromosomal form, but that's fine. So the DNA is unzipped. We go to the gene we want. So let's say this code right here codes for a certain gene. Let's say this codes for, let's say the codes for, I don't know, making a red blood cell or something like that, okay? So let's say the body needs a hemoglobin. Let's say it's hemoglobin for the red blood cell. So the body needs some hemoglobin. So it's going to go in your bone marrow. You're going to have some stem cells. And it's going to go to that specific gene. And this right here, the RNA polymerase, is going to run across these nucleotides on the DNA strand. And it's going to kick out an mRNA strand. In this mRNA strand, messenger RNA right here, that is where, that's your code that's going to go to your ribosomes and make the protein hemoglobin. I think that's a protein. Okay, this example. All right, so that's how transcription is done. Here's another version of it. I don't think this one's as good, but we see that the DNA is unzipped. We see that we have a mRNA strand being created from one of the DNA strands. Okay, so that's